Thanks so much for this opportunity to present some of our recent work and generally the state of the field, understanding immune responses to cancer immunotherapy from outside of the tumor and how those really impact therapeutic efficacy. My name is Matt Spitzer. I'm an associate professor at UCSF and the Gladstone Institute, an investigator of the Parker Institute and the Chan Zuckerberg Biohub. A few financial disclosures. I'm a shareholder and co-founder of Teiko Bio, which is a company that provides immune monitoring services. And I've also received research support from Genentech, who's the manufacturer of atezolizumab, which will be discussed in the presentation today. About work that we've done more recently to evaluate to what extent this is true in patients and whether the same types of CD8 T cells are responding. So this was just recently published in Cell. And what we did was to take patients with head and neck squamous cell carcinomas and to analyze paired tumors and lymph nodes from these individuals. And we did this using mass cytometry, using an antibody panel oriented toward T cell differentiation states. We performed dimensionality reduction on all of the CD8 T cells and identified clusters of CD8 T cells that express shared protein expression profiles, and then annotated these based on their differentiation state as indicated here in the heat map. And we found populations of progenitor-exhausted, terminally exhausted cells, as well as the more conventional differentiation states, naive central memory, effector memory, and tissue-resident memory cells, and asked which of these is more or less abundant in the lymph nodes compared to the tumors from these paired samples from the same individual. So we performed a differential abundance analysis on these clusters and identified that many of the terminally exhausted clusters were enriched in the tumor, and in contrast, the progenitor-exhausted clusters, 14 and 8, were enriched in the uninvolved lymph node. So these formed a larger percent of the antigen-experienced T cell pool within the uninvolved lymph node than they did in the tumor, as quantified on the right. These were highly statistically significant. We then wanted to know, are these progenitor-exhausted cells in the lymph node clonally related to cells that we see in the tumor microenvironment? And so we analyzed CD8 T cells from 5 additional patients by single-cell RNA sequencing paired with T cell receptor sequencing. We identified the CD8 T cells, clustered them, and colored them in this UMAP dimensionality reduction plot based on their tissue of origin, identifying regions that were enriched in lymph node or tumor respectively. And we overlaid onto this gene signatures from a recent meta-analysis of CD8 T cells across many different human cancers published by Zheng et al. And we're able to see regions that were enriched for the Tpex or TCF7 positive populations compared to the terminally exhausted populations, which were enriched over on the right, where cells from the tumor microenvironment were also preferentially enriched. We then identified clones based on their shared T cell receptor sequences, and were able to identify across each patient clones that were shared between the lymph node and the tumor. And we asked what were the differentiation states of these CD8 T cells that belonged to these shared clones across each tissue. We found that the cells in the lymph node were more likely to express the TCF7 Tpex-like exhaustion score, so progenitor-exhausted cells, whereas their counterparts with the same clones, same T cell receptor sequences belonging to the same clones, were enriched in the terminally exhausted cells in the tumor microenvironment. And plotting this on a clone-by-clone basis, we also saw that the cells in the lymph node had overweighted Tpex gene expression signatures relative to the terminally exhausted signatures compared to the counterparts that belonged to the same clone but were in the tumor microenvironment instead. Now, of course, we wanted to know, do these cells respond to checkpoint inhibitor treatment? And so here we, together with our clinical collaborators at UCSF, performed a small neoadjuvant clinical trial where patients with head and neck cancer were treated with a single cycle of atezolizumab, anti-PD-L1, shortly before surgery. And in the context of this study, we were able to look at what happens in the lymph nodes after patients are treated with this checkpoint inhibitor, and we compared it to lymph nodes collected from 22 patients receiving standard of care surgery without immunotherapy. We identified regions in these lymph nodes and put them into a tissue microarray that were analyzed by multiplexed ion beam imaging. And multiplexed ion beam imaging is a technology related to mass cytometry using metal conjugated antibodies to quantify proteins. But instead of doing this on a single cell basis on cells in liquid suspension, it instead uses a primary ion source to liberate these ions from antibodies that have been used to stain intact tissues, intact tissue section. And so this allows us to see where physically within the tissue each cell is localized. And it's compatible with formalin-fixed paraffin-embedded tissues that are oftentimes the way that tissues are processed for clinical pathology applications. And so we did the same thing from our MIBI data. We identified all of the CD8 T cells, and we annotated them based on their differentiation states, based on the proteins they express, and found that a population of Tpex was present in the lymph nodes, as well as some more intermediate exhausted cells that had downregulated expression of the transcription factor TCF1, but had not yet upregulated the expression of inhibitory receptors like TIM3 or CD39 associated with a terminally exhausted state. And we asked across all of these CD8 T cell clusters, how do they change after treatment with immunotherapy? And we found that the progenitor exhausted population, cluster 18, was the most significantly reduced in patients that had recently been treated with anti-PD-L1, whereas cluster 16, this intermediate exhausted population, was amongst those that had the largest increases in their abundance within these lymph nodes. So we quantified this more directly, and we found a strong trend toward a decrease in Tpex in immunotherapy-treated lymph nodes from patients, as well as a trend toward an increase in the intermediate exhausted cells. And if we express this as a ratio of the PD-1 positive CD8 T cell population, we could see a significant skewing of this population toward the more differentiated intermediate exhausted state as opposed to the progenitor exhausted state. And because this was spatial data, we were also able to look at which cells are physically neighboring Tpex in the tissue, and found that Tpex after treatment were surrounded by larger numbers of these intermediate exhausted CD8 T cells. We then asked, do Tpex co-localize with any antigen-presenting cell populations that may be driving their activation after treatment? And indeed, we found that after treatment, Tpex were more likely to be surrounded by larger numbers of dendritic cells compared to patients that did not receive immunotherapy. And importantly, the total number of dendritic cells, or the percent of dendritic cells in these tissues, was not changed by treatment. So this really reflects a change in the localization of these cells. And moreover, we saw that the Tpex, as well as the intermediate exhausted cells that were neighboring dendritic cells, upregulated the expression of PD-1 in the lymph nodes from patients treated with checkpoint inhibitors, consistent with the activation of these cells. Now, as I mentioned, several studies had found that expansion of particular effector CD8 T cell populations in peripheral blood was associated with clinical benefit from immune checkpoint inhibitors. And so we were interested in understanding how these responses that we observed in the lymph node related to responses we might see in peripheral blood. So from the same patients, we collected pre- and post-treatment blood samples, which were analyzed together with some resected tumor tissues by mass cytometry. We identified the CD8 T cell clusters, subjected them to clustering, and annotated the identity of each cluster based on the expression profiles of the cells within that cluster. And what we found was that comparing the pre-treatment time point, baseline, to the post-treatment time point at the time of surgery after a cycle of checkpoint inhibitor immunotherapy, that there were two clusters of intermediate exhausted cells with similar phenotypes to those we saw expanding in the lymph node that also expanded in peripheral blood. We also saw that one of these clusters, cluster 5, significantly increased its proliferation in peripheral blood as measured by a Ki67 expression. So these cells had recently or were in the process of dividing. And we also saw this to be true of a TEMRA population in the peripheral blood. Now, it was possible that these cells in the blood originated from the lymph node CD8 T cells that we saw responding, or could have potentially egressed from the tumor. And so we asked, do we see evidence of similar populations proliferating in the tumor? And we did not. So these cells were Ki67 positive in the blood, but not in the tumor tissue, which would be consistent with their etiology as having come from the lymph node. And so this was really interesting to us because it really drove home this point that the responses we see in the lymph node are indeed associated with these detectable responses in peripheral blood, which is a great opportunity for immune monitoring. Now, the last question we wanted to address in this study is what happens in metastatic lymph nodes? Cancer cells can metastasize to regional lymph nodes, and we wanted to know if that impacts the responses of CD8 T cells within the lymph nodes when immunotherapies are used to treat patients. And from a number of patients, we were able to collect paired, uninvolved, and metastatic lymph nodes from the same individuals at the same time, draining the same primary tumor. And we asked what CD8 T cell clusters were differentially abundant between them. Now, these metastatic lymph nodes really looked a lot like the primary tumors. So they had a significant reduction in the abundance of these Tpex clusters as compared to the uninvolved lymph nodes from the same individuals. And we then asked whether these changes in the lymph node would extend to the response to immunotherapy. So we investigated metastatic lymph nodes from patients treated as part of the clinical trial, and we found that indeed, in metastatic lymph nodes, progenitor exhausted cells failed to be surrounded by larger numbers of intermediate exhausted cells, consistent with their lack of differentiation. And the Tpex and intermediate exhausted cells also failed to upregulate PD-1, consistent with the lack of activation of these cells in the metastatic lymph nodes compared to the uninvolved lymph nodes. Then we were curious what cell types these cells may be interacting with within the metastatic lymph nodes, and if they had a different phenotype. And indeed, we found that the dendritic cells surrounding the Tpex in these metastatic lymph nodes had upregulated many immunosuppressive molecules, including CD39, IDO, PD-L1, and TIM3, as compared to the dendritic cells from the uninvolved lymph nodes of recently treated patients. And moreover, we found that the Tpex were also surrounded by Tregs that had much higher FOXP3 expression and higher expression of TIM3, which are both consistent with the suppressive capacity of Tregs within these metastatic lymph nodes. And this is visualized in the representative image here at the right. So having seen that the response to checkpoint inhibitors is impaired in metastatic lymph nodes, we then wanted to know, is this then associated with a change in the detectable CD8 T cell response in peripheral blood? And indeed, that was what we saw. So patients with metastatic lymph nodes tended to have smaller increases in these intermediate exhausted cells within the lymph node and less proliferation of these cells after treatment. And so just to summarize the findings of our new study, what we've found here is that Tpex are indeed present in regional lymph nodes in human cancer patients, and these Tpex are clonally related to terminally exhausted cells that are present within the tumor microenvironment. We found that immune checkpoint blockade reduces Tpex abundance in these uninvolved lymph nodes, but increases the abundance of intermediate exhausted cells. We found that checkpoint blockade also results in Tpex localizing near DCs where they become activated. And we found that in metastatic lymph nodes, Tpex are both reduced in frequency and localized in immunosuppressive niches that prevent them from getting activated well when patients are treated with immune checkpoint inhibitors. And so with that, I'd really like to acknowledge everybody in the group that contributed, especially the first authors of the study, Trainalina Okholm, a fantastic computational biologist in the lab, Kyle Jones, a pathologist, and Maha Rahim, an exceptional experimental biologist. This was really a team effort that involved many people throughout the lab. Our clinical collaborators, Alan Al-Ghazi, who's the medical oncologist who treated these patients, and the PI on the clinical trial, Patrick Ha, the surgical oncologist who operated on all of these patients, Larry Fong, the director of our cancer immunotherapy program at UCSF, and the driver of many of these immunotherapy studies, Mika Van Zant, our fantastic collaborator in pathology, who really helped us access these lymph node tissues in a way that was consistent with and commensurate with the clinical pathology that needed to happen, Max Krummel and Alexi Combs, who were instrumental in the single-cell RNA sequencing studies, Stanley Tamaki, instrumental in the MIBI studies, Alex Chung, instrumental in study logistics, sample processing, collection, and Bushra Samad and Vrinda Johri, who helped with the single-cell RNA sequencing studies as well. Our fantastic collaborators at Stanford, Mike Angelo and Candice Liu, who helped with the MIBI analysis, Bill O'Gorman at Genentech, who was instrumental in setting up this program, and of course, most importantly, all of the study participants and their families, without whom and without whose participation and support and partnership, we would never be able to do these types of translational research studies, as well as all the funding sources that make these types of studies possible. Thank you so much. Particularly to Genentech and the Cancer Research Institute for their partnerships in this study, as well as the Parker Institute for Cancer Immunotherapy. And thank you so much for your attention. Take care, everybody.